Welcome biology students. This is Miss Teslin and I'm making a quick video for you to help with 2.3 Model Meiosis assignment. So here we are at the home page. We're going to course activities as before. We're going to go to lessons instead of assignments, although you could go find it through assignments as well. When we go to the lessons, as we click down or scroll down, I should say, we've got the 2.3 meiosis, and then we go right to clicking on this assignment. Meiosis is when animals and plants reproduce, where there's a genetic information exchange. So we call that sexual reproduction. So when you have the genetic information exchange, you can have all sorts of interesting new traits that appear. And that's a little bit of what we're going to be learning further on in terms of characteristic traits and evolution and all sorts of interesting things that way. But right now we're learning at the cellular level what is actually happening. Just like mitosis, meiosis has different phases, and they're actually called the same things, but they happen a little bit differently. So instead of with mitosis, where you have two identically genetically similar cells at the end to the original mother cell, so two identical daughter cells, when there is meiosis, you actually have four very different genetic information in the cells at the end. So instead of two daughter cells, you're going to have four cells that are not genetically identical. All right. So all of that said, this is going to be 18 points, just like the mitosis model. You're going to be using beads and string or whatever other information you want, and you're going to take pictures at each of the phases, or you can make a video as you go through the phases, moving things around. This clicking on here will show you, will take you into the lesson slides, and it actually explains. Here it gives you a little visual. You can click on this and make it bigger, and you can actually see what is happening. So like I said, for meiosis, you start out with this, and you get four genetically different cells at the end. Going back to this, you're going to include interphase in this section. As I mentioned, there's two parts: prophase 1, metaphase 1, anaphase, telophase 1, and cytokinesis, when the cells split the first time. And that's right here. That's splitting. And then both of these two new cells that are genetically different will go through prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis, where they split off into four. So each of the two cells split off into two more cells. Again, all genetically different. Okay. Clicking on this activity is going to take you to something that you can actually print out and follow along. And this is actually step by step what you would be doing. So for interphase, you can take a chalk or a marker that you can erase, an erasable marker. You draw a line, that can be your cell, and you can draw another circle, and that can be your nucleus. And then you have your blue beads and your red beads on the string, and you want to double them up because the DNA has doubled up. And then you're going to start into prophase, where you exchange the genetic material. So you're going to have a mix of red and blue beads in both the chromosomes. And then you go through metaphase, anaphase, telophase, and cytokinesis. You get two cells that are genetically different with, then they go through prophase, metaphase 2, anaphase 2, telophase 2, and cytokinesis at the very end. You're going to redraw those nuclear membranes through each of them, and each of them have the singular chromosome, so that you have four cells, four daughter cells. And then that's how you would model it. So it can either be several pictures, or it can be one video that you send to me, and that's it. Good luck.